Hello, my name is John Paul, and today I'll be presenting on sickle cell disease and how CRISPR gene editing could be used as a possible treatment. This project explores the biology behind the disorder, how the mutation affects the body, and the ethical questions that come with genetic engineering. Sickle cell disease is a genetic disorder that affects millions of people worldwide. I chose this topic because it's a perfect example of how a tiny change in DNA can lead to serious health problems. In this presentation, I'll explain how the disease is inherited, what mutation causes it, how it affects gene expression, and how CRISPR could potentially fix it. I'll also discuss some ethical concerns that come with editing human DNA. What is sickle cell disease? Sickle cell disease affects the shape of red blood cells. Instead of being round and flexible, the cells become stiff and curved, or sickle shaped. These sickle cells can't move smoothly through blood vessels. They get stuck, blood, block blood flow, and break apart easily. That's why people with this condition often experience pain, crisis, anemia, and sometimes organ damage. All of this starts from one mutation in the hemoglobin gene. Sickle cell disease is inherited in an autosomal recessive pattern. The mutation is located on chromosome 11 in the HBB gene. A person needs two copies of the sickle allele, one from each parent, to have the disease. If someone has only one copy, they're a carrier. Carriers don't usually have symptoms, but they can pass the allele onto their children. This is why understanding inheritance patterns is important when studying genetic disorders. When two carriers have a child, there's a 25% chance the child will completely be normal, a 50% chance the child will be a carrier, and a 25% chance the child will have the disease. This pattern would show up clearly in a pedigree chart. You can see how the disorder appears only when both parents pass down the sickle allele. This helps explain why the disease seems to skip generations sometimes. The DNA mutation. The entire disorder comes from a single base change in the HBB gene. Normally, the DNA codon is GAG, which codes for the amino acid glutamic acid. In sickle cell disease, that codon becomes GTG, which codes for valine instead. It's amazing how one small letter change in the DNA can completely change the shape and function of a protein and eventually affect someone's entire life. DNA, RNA to protein. Here's what happens next. The mutated DNA gets transcribed into mutated mRNA. So instead of the normal GAG codon, the mRNA has a GUG. When the ribosomes translates it, it puts valine in the hemoglobin protein. That one amino acid change makes hemoglobin molecules stick together under low oxygen. When they clump when they clump up, the red blood cell changes shape. These sickled cells can't carry oxygen well. They break apart easily and they get stuck in blood vessels. This is why the symptoms can be so severe. Even though every cell in the body has the Even though every cell in the body has the HBB gene, it's only turned on in developing red blood cells. That's because of gene regulation. Different genes turn on different cell types. In sickle cell disease, the mutation doesn't stop the gene from being expressed. The body is still making hemoglobin, but it's making the faulty version. Understanding gene regulation is also important for treatments because some therapies try to increase fetal hemoglobin, which doesn't sickle. Why CRISPR is needed. Current treatments for sickle cell disease mostly manage symptoms, but they don't fix the actual genetic cause. CRISPR is exciting because it can target the exact mutation that causes the disorder. Instead of treating the pain crisis or the anemia, CRISPR aims to correct the DNA itself. That opens the possibility of giving patients a long-term or even permanent solution. CRISPR Cas9 steps. Here's how CRISPR treatment would work. First, doctors collect stem cells from the patient's bone marrow or blood. These are the cells that make all future red blood cells. Then scientists design a guide RNA that leads the Cas9 enzyme to the exact GTG mutation in the HBB gene. Cas9 cuts the DNA at the location. The cell is given a correct DNA template with the normal GAG sequence. The cell repairs the cut using that template. After the edit, the corrected stem cells are put back into the patient. If everything works, those corrected cells start producing healthy red blood cells. Potential benefits. If CRISPR works, the benefits would be life-changing. Patients may have fewer pain crises, fewer hospital visits, and better oxygen levels. Their red blood cells would work the way they're supposed to. It could dramatically improve their quality of life. And because the change is made at the DNA level, the benefits could last for years or even permanently. Ethical concerns. Even though CRISPR has huge potential, there are ethical concerns. One issue is off-target effects. CRISPR might accidentally edit the wrong part of the genome. Another issue is access. Gene editing treatments can be extremely expensive, which could leave out many communities who need them the most. Some people also worry that genetic engineering could eventually be used for non-medical purposes, like choosing traits in children. And because these edits may last a lifetime, we have to be sure they're safe. These questions show why bioethics is a major part of genetic engineering. Broader health impact. If CRISPR treatment becomes safe, effective, and affordable, it could help millions of people around the world. Sickle cell disease is especially common in Africa, India, and the Middle East. Fixing a single mutation could dramatically reduce suffering in those regions, and success with sickle cell disease could lead to treatments for other disorders caused by single gene mutations. Monitoring success. To make sure CRISPR worked, doctors need to check DNA, RNA, protein levels, and the patient's symptoms. They can sequence the DNA to see if the GTG mutation has been corrected back to GAG. They can measure RNA transcripts to ensure the right mRNA is produced. They can test hemoglobin levels to see if normal hemoglobin is present. They can also check the shape of red blood cells to confirm sickling has decreased. And of course, they can keep track of whether the patient feels better. In conclusion, sickle cell disease is caused by a single mutation, but it has a huge effect on the body. CRISPR offers a way to fix that mutation at the source, potentially curing that disease. But because we are editing human DNA, we have to think carefully about safety, access, and ethics. This topic shows how powerful genetic engineering is and why responsibility is so important in science.